Welcome back to the Break It Down series. This is episode 2 and the topic for this episode is biological molecules. If you are new to this channel, you will not be disappointed. Subscribe to be a part of the Bio Squad, like and watch the rest of the videos too. Let's get started then, shall we? Question 1. A student tested 4 samples of food A, B, C and D for the presence of lipids, protein, reducing sugars and starch. One of the food samples, milk, was found to contain lipid, protein and reducing sugar. Which of the food samples shown in the results below is milk? The food sample, which is milk, only contains lipid, protein and reducing sugar, whereas no starch is present. And what's the test for starch? Well, the test for starch is adding iodine in potassium iodide solution. Therefore, the result for starch should be a negative result. And if we go through the answers A, B, C and D, only A and D shows a negative result. Whereas blue-black is the positive test result for starch. However, starch is not present, leaving us with the answer being either A or D. They have mentioned that lipids, protein and reducing sugars are present. So, if reducing sugar is present, when you boil it with Benedict solution, it should not be blue, but rather it is a color scale from blue, green, yellow, orange and finally red. Blue being where there is no reducing sugar and red being where there is the most reducing sugar. However, in option D, they have mentioned blue for boiling with Benedict solution. And since reducing sugar is present, the answer D is incorrect because it shouldn't be blue but either any other color from green, yellow, orange and red. Therefore, answer A is the correct answer. A. Adding Biuret reagent, lilac. Yes, that is correct because the protein positive test is purple or lilac. Adding iodine in potassium iodide solution, orange. Yes, that is correct because no starch is present. Boiling with Benedict solution, orange precipitate. Yes, that is again correct. Mixing with ethanol and adding to water. The positive test result for lipids is milky emulsion. Therefore, A is the correct choice. Question 2. Which statement describes how the molecular structure of starch is suited to its function? A. Amylose has a branch structure and amylopectin is coiled to give a compact molecule. That is incorrect because amylose is not a branched structure whereas amylopectin is branched. Therefore, A is incorrect. B. In the breakdown of amylose and amylopectin, many hydrolysis reactions release stored energy. They are asking how is starch suited to its function. And in plants, energy release is not the main purpose of starch, however, to regulate osmotic properties. Therefore, B is incorrect. C. In the formation of amylose and amylopectin, many condensation reactions allow the release of stored energy. That is again incorrect because energy is released not by condensation reactions, however by hydrolysis reactions. And again, the main function of starch is to regulate the osmotic properties and not to release stored energy. Therefore, C is again incorrect. D. The final amylose-amylopectin complex is insoluble and does not affect the osmotic properties. Yes, that is correct. Amylopectin and amylose, they are polymers and they do not affect the osmotic properties as they are insoluble. Only soluble molecules will affect the osmotic properties. Therefore, D is the correct answer. Question 3. Which diagram represents part of the ring form of a molecule of beta-glucose? In beta-glucose, the OH group or the hydroxyl group lies above the plane. If it lies above the plane, that is beta-glucose. And if you go through the answers A, B, C and D, answers A and D both show the hydroxyl group on the first carbon lying above the plane. Hence, considering between answers A and D. Answer D is obviously incorrect because in answer D there are 2 OH groups lying side by side. However, in a molecule of glucose, there shouldn't be any 2 OH groups lying side by side at carbon 1 and at carbon 4. Therefore, D is incorrect, leaving us with the answer being A. Question 4. How many fatty acid residues are normally present in a phospholipid molecule? A phospholipid has 2 fatty acid tails instead of 3 because one fatty acid is replaced by a phosphate group. Therefore, the answer should be B, 2 fatty acid tails. Question 5. Which feature distinguishes starch from glycogen? A. Starch contains alpha glucose. B. Starch contains 1,6 glycosidic bonds. C. Starch has an unbranched component. D. Starch is a polysaccharide. Both glycogen and starch is a polysaccharide, therefore D is incorrect. C. Starch is an unbranched component. Yes, that is correct because starch has amylose, which is an unbranched component, whereas in glycogen is a branched molecule or a branched structure. It has both 1,4 and 1,6 linkages and doesn't have any unbranched components. Therefore, the answer C does distinguish starch from glycogen. If you look at B, starch contains 1,6 glycosidic bonds. Well, even glycogen contains 1,6 glycosidic bonds, therefore it doesn't distinguish the two of them. A, starch contains alpha glucose. Yes, even glycogen contains alpha glucose, therefore it doesn't show any difference between the two. Hence the answer is C. Question 6. The diagram shows part of a carbohydrate molecule. If all the 1,4 glycosidic bonds in this molecule are hydrolyzed, how many water molecules will be used and how many separate glucose molecules will be produced? I have underlined where hydrolysis will occur, and only 2 water molecules will be required, as hydrolysis occurs between the 1,4 glycosidic bonds, as they have mentioned in the question, if all the 1,4 glycosidic bonds are hydrolyzed. And I have already shown where 1,4 glycosidic bonds occurs in this molecule, underlined in red. And if you hydrolyze them, 2 water molecules will be required, 1 for each underlined part. Therefore, if you go to the answers, number of water molecules used should be 2, leaving us with the answer being B. And number of glucose molecules produced is also 2. Yes, that is correct, because if you hydrolyze all the 1,4 glycosidic bonds in this molecule, you won't get 3 glucose molecules, because the center or the middle molecule is not a glucose molecule, as it has a branched component attached above it. Therefore, it won't be glucose. Hence, the answer is B. Question 7. A peptide bond is formed between atoms of which 2 elements? Peptide bonds occurs only in proteins between 2 amino acids, and hence will form between the atoms carbon and nitrogen. Therefore, the answer is C, carbon and nitrogen. Question 8. Which features affect the tensile strength of collagen? 1. The helical structure of collagen chains. 2. The small R groups of the amino acids in collagen. 3. The insoluble nature of collagen. 4. The covalent bonds between collagen molecules. Feature 3 doesn't affect the tensile strength. It doesn't affect the strength but rather the osmotic properties. Hence 3 is incorrect. If you look at 1, 2 and 4, all the features apart from 3 are correct, because 1 aids in the tightly packing of molecules. Feature number 2, the small R group, it allows the chains to be packed closely together, hence increasing the tensile strength. 4. The covalent bonds between collagen molecules. Yes, that again leads to a strength of collagen, because it holds the neighboring collagen molecules together. Therefore, the answer is C, 1, 2 and 4 only. Question 9. When red cells leave the lungs, hemoglobin is 98% saturated. How many molecules of oxygen will be carried by the majority of hemoglobin molecules? If you recall correctly, a hemoglobin has 4 heme groups, therefore can carry maximum 4 oxygen molecules. Hence, since they are asking how many molecules of oxygen will be carried, and the keyword over here is molecules, that will be 4, as only 4 oxygen molecules can be carried, that is 8 oxygen atoms. Therefore, the answer is B, 4 oxygen molecules. Question 10. Which rows show the chemical groups present in the biological molecules listed? If you go to the rows, row number 3 is obviously incorrect, because if you look at presence of 2 or more hydroxyl groups, they have mentioned no. However, it should be yes, because the glycerol molecule has 3 OH groups. Therefore, row number 3 is obviously incorrect, and if you go to the answers A, B, C and D, all A, C and D has 3 in it except B. Therefore, the answer should be B, 1, 2 and 4. Let's go to 1. Amino acid, presence of carboxyl groups, yes. Presence of 2 or more hydroxyl groups, no. That is correct, because all acids have carboxyl groups and do not have any hydroxyl or alcohol groups. Therefore, 1 is correct. 2. Beta glucose, no carboxyl groups. Presence of 2 or more hydroxyl groups, yes. Yes, that is correct, because beta glucose has many hydroxyl groups. It has on carbon 1, carbon 2, carbon 3 and so on, and it doesn't have any carboxyl groups. Therefore, 2 is also correct. Let's look at 4. Fatty acid, yes, carboxyl groups should be present, as all acids have a carboxyl group, and no hydroxyl groups. Therefore, 1, 2 and 4 is correct, leaving us with the answer being B. Question 11. Some foods contain hydrogenated vegetable oils. These are unsaturated fats that have been converted to saturated fats. Which property of the fats will have changed? Unsaturated fats have double bonds, hence they are more fluid. If they are converted to saturated fats, they will have no double bonds, hence will have less fluidity. And less fluidity means their hydrocarbon chains will fit together more closely. Therefore, which property of the fats will have changed? It should be A. The hydrocarbon chains will fit together more closely. Let's look at B. Their solubility in water will increase. That is incorrect. Since the fluidity decreases, the solubility in water can't increase. C. They will have more double bonds in their molecules. That is incorrect because unsaturated has double bonds whereas saturated doesn't have double bonds. Therefore, it cannot have more double bonds. Hence, C is incorrect. D. They will remain liquid at room temperature. Only presence of double bonds will cause it to be more fluid, hence remain liquid at room temperature. However, if they are changed to saturated fats where there are no double bonds, hence less fluidity, they will become solids at room temperature, not liquids. Therefore, D is again incorrect, leaving us with the answer being A. Question 12. Which molecular bonds will be broken by hydrolysis when a molecule of glycogen is converted to glucose? 1,2 bonds. Well, 1,2 bonds are not affected by hydrolysis at all. It's only 1,4 and 1,6 because glycogen has 1,4 and 1,6 linkages. Therefore, only 1,4 and 1,6 linkages will be affected by hydrolysis, not 1,2. Leaving us with the answer being B. 1,2 is unbroken. 1,4 and 1,6, yes, it is broken. Question 13. Which correctly matches the functional and structural features of cellulose, collagen, glycogen or triglyceride? Key thing to note is that cellulose and collagen are both fibrous as well as unbranched, and cellulose is there for structural purposes or structural functions such as support in cell walls of plants. Also, cellulose molecule and collagen molecules are both held together by hydrogen bonds within the molecule. Hence, if we consider options A, B, C and D, option A, cellulose, function support, yes that is correct. Fibrous, yes that is correct. Molecule held together by hydrogen bonds, that is also correct. Branch chains. And if you look at triglyceride energy source, fibrous, yes that is false. Molecule held together by hydrogen bonds, yes that is also false. Branch chains, yes again that is also false. Therefore, A is the correct answer. Why are B, C and D incorrect? If you look at B, for cellulose, they have put false under molecule held together by hydrogen bonds. However, cellulose does have hydrogen bonds holding the molecule together. Therefore, B is incorrect. Let's look at C. For collagen under branch chains, they have put true. However, collagen doesn't have any branch chains. Therefore, C is incorrect. Let's look at D. Under glycogen, they have mentioned molecule held together by hydrogen bonds. However, for glycogen, only glycosidic bonds are present and no hydrogen bonds are involved at all, leaving us with the answer being A. Question 14. Tests were performed on samples from a mixture of biological molecules. When iodine in potassium iodide solution was added to a sample, the mixture turned black. When the biuret test was carried out on another sample, the mixture turned purple. Which biological molecules were in the mixture? If the mixture turned black when adding iodine in potassium iodide solution, that means starch is present, because blue-black is the positive test result for starch. And when the biuret test is carried out, the test result is purple, that means proteins are present. So if you consider the answers A, B, C and D, the best and possible answer is A, amylase and starch, because yes, starch should be present and amylase is an enzyme and all enzymes are made up of proteins. Therefore, A is the correct answer. Why can't we consider B? Because B mentions cellulose and cellulose is not a protein as it has glycosidic bonds. Therefore, the answer is A. Question 15. Which of the statements about polysaccharides can be used to describe both amylopectin and cellulose? 1. Adjacent glucose molecules are rotated by 180 degrees. 2. Contains 1,4-glycosidic bonds. 3. Polymer of alpha glucose. Cellulose is made up of beta glucose and not alpha glucose. Therefore, 3 cannot be used to describe cellulose. Hence, it doesn't describe both amylopectin and cellulose. Let's look at 2. Contains 1,4-glycosidic bonds. Yes, that is correct because amylopectin does have 1,4-glycosidic bonds and so does cellulose. Let's look at 1. Adjacent glucose molecules are rotated by 180 degrees. That is only correct for cellulose and not amylopectin. Hence, it doesn't describe both amylopectin and cellulose, leaving us with the answer being A, 2 only. Question 16. Which molecule in the key is sucrose? Key thing you should know is that sucrose is a disaccharide and hence is a non-reducing sugar. Therefore, is a reducing sugar? That should be no. And contains hexose sugar? Yes, because sucrose is a 6-carbon sugar unit. Therefore, the answer is C. Is a reducing sugar? No. Contains hexose sugar? Yes. Question 17. Which molecules have a structural formula that contains C double bond O bonds? 1. Fatty acids, 2. Glucose, 3. Glycerol. Glycerol is an alcohol molecule and hence will have no carbon double bond oxygen bonds. Therefore, answers B, C and D are incorrect as it has 3 in it, leaving us with the answer being A, 1 and 2 only. Fatty acids, yes, because it has a carboxyl group, hence will have C double bond O. Glucose also does have in the linear structure. Question 18. Heating with which solution breaks glycosidic bonds? Breaking of glycosidic bonds is hydrolysis, and hydrolysis can be done in different ways. It can be done by using water or it can be done by acid hydrolysis. Therefore, the best possible answer over here is B, dilute hydrochloric acid. Question 19. Diagram shows 4 molecules. Which 2 molecules condense to form sucrose? Sucrose is a disaccharide which is made up of alpha glucose and fructose, and S represents sucrose and P represents alpha glucose. Why is R not alpha glucose? Well, in R, if you look at the first carbon atom, the OH group lies above the plane, and if the OH group lies above the plane, that is beta glucose, not alpha glucose. If only the OH group lies below the plane, then that is alpha glucose. Hence, P is alpha glucose. And if you study the structure of sucrose properly, Q cannot be sucrose, as in Q there are 2 OH groups lying side by side, which is incorrect. If you look at carbon 2 and carbon 3, there are 2 OH groups lying side by side, whereas in S there isn't. Therefore, S is sucrose. Therefore, the answer is B, P and S only. Question 20. Hemoglobin consists of 2 alpha chains and 2 beta chains. Approximately 5% of all humans have one amino acid in the beta chain different from normal. Which levels of protein structure could be changed in these humans? In these answers, tertiary structure is a key point. It should be present in the answer because tertiary structure determines the overall shape and function of the protein. Hence, if one amino acid changes, then it will change the function and will lead to a change in the tertiary structure because tertiary structure is related to the function. Therefore, the answer is C, primary, secondary and tertiary only, because only C has tertiary involved in the answer, whereas A, B and D doesn't. Therefore, C is the correct answer. Question 21. Which of the statements about polysaccharides can be used to describe both glycogen and amylopectin? 1. Contains 1,4-glycosidic bonds. 2. Contains 1,6-glycosidic bonds. 3. Polymer of alpha glucose. Both glycogen and amylopectin are polymers and both will have 1,4 as well as 1,6 linkages because glycogen is an unbranched molecule and amylopectin is an unbranched component. Hence, 1,6 linkages means that it is unbranched. Therefore, since it has both 1,4 and 1,6 glycosidic bonds, statement 1 and 2 should be correct. If you look at 3, polymer of alpha glucose. Yes, both glycogen and amylopectin are polymers of alpha glucose. Hence, the answer is D, 1, 2 and 3. Question 22. The R groups, side chains of amino acids can be hydrophobic, hydrophilic, acidic or basic. The diagram shows 4 different amino acid R groups. Which row is correct for these amino acid R groups? Remember that I mentioned in one of the previous questions that all acids do have a carboxyl group, that is COOH. Hence, in order to be acidic, there should be a COOH group, leaving us with the only answer being A, because under acidic they have mentioned the correct amino acid as ASP, which is the only R group that has a COOH group. Therefore, A is the correct choice. Hydrophobic is ALA, hydrophilic is SER, acidic is ASP, basic is LYS. Question 23. Which describes the emulsion test for the presence of lipids? Over here, you simply have to know how to do the emulsion test. First, you should add ethanol, then pour into water, and finally you should shake. If lipids are present, then it will lead to a milky emulsion. Therefore, the answer is B. Add ethanol, pour into water and shake. Question 24. The diagram shows part of a molecule of glycogen. How does a molecule differ from a molecule of amylose? Well, amylose is an unbranched component, whereas glycogen is branched. And if a molecule is branched, it should definitely have 1,6 linkages. Whereas amylose is unbranched, therefore doesn't have any 1,6 linkages. It only has 1,4 linkages, and that is also of alpha glucose. Both glycogen and amylose are made up of alpha glucose. But since the question asks how does the molecule differ from a molecule of amylose, that should be A. Amylose has only alpha-1,4 glycosidic bonds, whereas glycogen has both alpha-1,4 glycosidic bonds as well as alpha-1,6 glycosidic bonds. Hence the answer is A. Why are B, C and D wrong? Well, B. Amylose has only beta-1,4 glycosidic bonds. That is incorrect because amylose doesn't have beta linkages. C. Amylose has alpha-1,6. That is incorrect. Again, 1,6 is related to branched structures or branched molecules, whereas amylose is an unbranched component. D. Amylose has beta-1,4. That is also incorrect because amylose is made up of only alpha glucose. Question 25. Which molecules contain a carboxyl group? All acids will definitely have a carboxyl group or a COOH group. Therefore, since only 1, 3 and 4 talks about acids, the answer should be C, 1, 3 and 4 only. Why is 2 incorrect? Because 2 is a alcohol molecule, so only hydroxyl groups are present and doesn't have carboxyl groups. Hence the answer is C, 1, 3 and 4 only. Question 26. The diagram shows a triglyceride molecule that has been partially hydrolyzed. What will be the products of the total hydrolysis of the molecule shown? I have marked where hydrolysis will occur. Hydrolysis occurs where there is a presence of an ester bond, and I have already marked where it is present. Therefore, if hydrolysis occurs there will be 2 molecules formed. And what are those 2 molecules? Well, a triglyceride has 3 ester bonds. However, in this molecule they have only shown 1 ester bond. Therefore, that will lead to 1 glycerol molecule and 1 fatty acid molecule being formed. How do we know which one it is? Whether is it unsaturated fatty acid or saturated fatty acid? You have to look at the presence of double bonds. If double bonds are present, that means it is unsaturated, as only unsaturated fatty acids will have double bonds. Saturated fatty acids have no double bonds. Therefore, the best possible answer is B, a molecule of glycerol and an unsaturated fatty acid molecule only. Why is C and D incorrect? Because C talks about a molecule of water being formed. However, in hydrolysis, a molecule of water is used, not formed. A molecule of water will only be formed in condensation reactions. Therefore, C and D are incorrect. And A is incorrect because it mentions saturated fatty acids, whereas over here it is unsaturated because of a presence of a double bond. Question 27. Well, if you recall correctly, I have already solved this question in one of the previous questions. Hence, the answer is A. You could refer to the explanation by looking at the previous questions. Question 28. The diagram shows a relationship between the levels of protein structure and bonds. Which row is correct? Key thing you should note is that peptide bonds are only present in primary structures, leaving us with the only answer being D, because all the other answers A, B and C has peptide in other levels of protein structure, which is incorrect. Peptide bonds are only involved in the primary structure. Therefore, the answer is D. The formation of a tertiary structure from a secondary structure may need ionic bonds, as also the formation of a quaternary structure from a tertiary structure. Therefore, D is a correct choice. Question 29. Which type of bond holds together an alpha helix or beta pleated sheet of a protein? Alpha helix or beta pleated sheet talks about the secondary structure, and secondary structure is mainly concerned with hydrogen bonds. Therefore, the answer is B, hydrogen. Why is A, C and D incorrect? Well, both A and C are involved in tertiary structures, and D, peptide. Peptide bond, as I mentioned before, is involved only with the primary structure. However, in this question they have mentioned alpha helix or beta pleated sheet, and that talks about the secondary structure. If you recall the definition of a secondary structure, it does involve alpha helix and beta pleated sheet. Question 30. The statements are about the properties of water. What allows a small insect to rest on the surface of a pond? The only possible answer over here is statement 1, because hydrogen bonds leads to high surface tension, leading to the small insect being able to rest on the surface of a pond. Hence the answer is C, 1 only. Statement 2 and 3 doesn't have anything to do with the insect being able to rest on the surface of a pond. Question 31. Which bonds hold together the structure of cellulose? 1 glycosidic, 2 hydrogen, 3 ionic. Cellulose doesn't involve any ionic bonds in it. Therefore, answers having bond 3 is incorrect, leaving us with the only answer being B, 1 and 2 only. Glycosidic bonds, yes, because cellulose is a polymer made up of beta glucose molecules, hence glycosidic bonds are involved. 2 hydrogen, yes, that is also involved because hydrogen bonds holds the neighboring cellulose molecules together. Therefore, B is the correct answer. Question 32. Which bonds are found in glycogen? Glycogen is a polymer made up of alpha glucose only. It doesn't have any beta glucose in it, therefore won't have any beta bonds. Hence, answers C and D are incorrect. If you look between answers A and B, glycogen is an unbranched molecule and all unbranched molecules do have 1,6 linkages. Hence, since glycogen is unbranched, it will have both 1,4 as well as 1,6 alpha bonds, leaving us with the answer being B. Question 33. Which diagram shows the bond linking the individual units of both cellulose and glycogen? Glycosidic bond is only represented by diagram D. Diagram A involves phosphate, which doesn't link the individual units of both cellulose and glycogen. B involves nitrogen, and nitrogen is involved in proteins. Therefore, B is incorrect. C is an ester bond due to presence of C double bond O. Hence again is incorrect. D. Yes, D is the correct answer as D is representing a glycosidic bond. Question 34. Which row describes a triglyceride? Triglycerides are hydrophobic in nature as they are non-polar as well as they are soluble in alcohol. Therefore, for hydrophilic it should be not correct and for soluble in alcohol it should be correct, leaving us with the answer being C. Question 35. The diagram shows the amino acid serine. Which circled area is different in all other amino acids? All amino acids will have 3 constant parts and they are a hydrogen atom, a COOH group and NH2. All amino acids have these 3 parts being constant, they don't change. The one that changes and hence leads to the difference between all the other amino acids is the R group, and the R group over here is represented by C. All the other answers A, B and D are incorrect because A has NH2 being different. However, NH2 is constant in all the amino acids. In B they have circled a hydrogen atom, whereas all amino acids do have a hydrogen atom. In D they have circled the carboxyl group or the C double bond O single bond OH group, which is constant in all amino acids, as acids will have a carboxyl group. Therefore, D is again incorrect. The only circled area that is different in all the other amino acids is the R group, which is represented by C. Question 36. Tests for biological molecules were carried out on 3 solutions. The observations were as follows. Solution 1. Benedict's test blue to orange. Solution 2. Benedict's test after acid hydrolysis blue to red. Solution 3. Biuret test blue to purple. Which observations would show the solutions that contain sucrose and amylase? If you recall one of the earlier questions we did, amylase is an enzyme and all enzymes are proteins. Therefore, solution 3 should be a correct observation, as Biuret test is carried out for proteins, and if it changes from blue to purple, that means it is a positive test result. Therefore, solution 3 should be a correct observation. And sucrose. Sucrose is a disaccharide and hence is a non-reducing sugar. How do you carry out the Benedict's test for non-reducing sugars? You should carry out acid hydrolysis first, then the Benedict's test. Therefore, solution 2 is also a correct observation. Solution 1 is not a correct observation as solution 1 is only carried out for reducing sugars, whereas sucrose is a non-reducing sugar. Hence the answer is C, 2 and 3 only. Question 37. A solution containing equal masses of amylose and amylopectin is completely hydrolyzed. Which molecules will be found after the hydrolysis? Amylose is an unbranched component, hence only has 1,4 linkages. Amylopectin is branched and has both 1,4 and 1,6 linkages. If you go to the answers A, B, C and D, it doesn't mention any of the linkages. Therefore, we should consider between the substance that makes up these polymers. Amylose and amylopectin are both made up of alpha glucose only. Therefore, the best possible answer is A, alpha glucose, as all the other answers B, C and D mentions beta glucose in it, which is incorrect. Question 38. Which statements about a triglyceride molecule are correct? 1. It always contains unsaturated hydrocarbon tails. 2. It is formed using ester bonds. 3. It does not form hydrogen bonds. 4. It contains a hydrophilic glycerol head. Statement 4 is obviously incorrect because it doesn't have a hydrophilic glycerol head. Instead, it has a hydrophobic glycerol head because triglyceride molecules are insoluble in water due to its nonpolar nature. Therefore, statement 4 is incorrect. If you look at statement 3, it does not form hydrogen bonds. Yes, that is correct. 2. It is formed using ester bonds. That is also correct. 1. It always contains unsaturated hydrocarbon tails. That is incorrect, because not always hydrocarbon tails are unsaturated. It could be saturated due to no presence of double bonds. Hence the answer is D. Statement 2 and 3 are the only correct statements. Question 39. The table shows some features of collagen and hemoglobin. Which row is collagen? Key thing you should know is that collagen is a fibrous protein and hemoglobin is a globular protein. Therefore, if you go to the answers A, B, C and D, type of protein should be fibrous. Hence the answer being either A or B. Collagen is also made up of 3 chains or 3 polypeptides lying side by side and is insoluble in water. Therefore, the correct answer is A. Question 40. The diagram shows 4 monosaccharides with the formula C6H12O6. Which diagrams do not show glucose molecules? The keyword over here is does not show glucose molecules. If you go to diagrams 1, 2, 3 and 4, diagram 1 and 2 are the correct representations of glucose molecules. Since they are asking do not show glucose molecules, it should be diagrams 3 and 4. How do we know which one doesn't show glucose molecules? If you look at diagram 3 and 4, at carbons 2 and at carbon 3, there are 2 OH groups lying side by side. However, glucose molecules do not have OH groups lying side by side at carbon 2 and at carbon 3. Therefore, the correct answer is D, 3 and 4, since the question refers to does not show glucose molecules. There, we have just covered around 40 questions in under 30 minutes. This is the end of this episode. Do leave a like, share with your friends, and put a comment down below on which episode I should be doing next. Thank you for making it till the end. Stay tuned for more.